Division of cells. In this video we are going to look at Introduction to cell division and chromosomes. Different stages in cell cycle. Stem cells. Plant meristem. Therapeutic cloning. Cell division. The process of cell duplication, or reproduction, is called cell division. This fundamental process allows living organisms to grow, develop, and reproduce. During cell division, a parent cell divides into two or more daughter cells, each containing a copy of the genetic material and cellular components necessary for independent functioning of the cell. This process ensures the continuity of life and the transmission of genetic information from one generation to the next. Chromosomes. Chromosomes are thread-like structures that carry a large number of genes. They are located in the nucleus of plant and animal cells. The structure of chromosomes includes a DNA molecule and proteins, forming a thread-like shape. Gene is the basic physical and fundamental unit of heredity. It is a section of a DNA molecule consisting of a sequence of bases that encode a specific protein and influence the characteristics of an organism. Though chromosomes exist as pairs in normal cells, there are some exceptions. For example, the human gamete, or reproductive cell, carries only one copy of each chromosome. A human body cell contains 2 copies of every chromosome. Cell cycle. The cell cycle refers to the sequence of processes that occur within a cell prior to its division, resulting in the formation of 2 identical daughter cells. There are 3 main stages in the cell cycle. Stage 1. During stage 1 of the cell cycle, the cell increases in size and undergoes DNA replication, resulting in the production of 2 copies of each chromosome. This stage is also referred to as the growth phase. The quantity of subcellular structures, like ribosomes and mitochondria, increases during this stage of the cell cycle. Stage 2. The stage 2 of the cell cycle is called mitosis. It is a special type of cell division in which one cell divides into 2 identical daughter cells. This is a process of cell duplication or reproduction. During this stage, the nuclear membrane disintegrates and the chromosomes align themselves in the middle of the cell. The 2 sets of chromosomes are then pulled towards opposite ends of the cell, leading to the division of the nucleus into 2 separate nuclei. The main function of mitosis in plants is for repairing, growing, and reproducing asexually. Stage 3. In stage 3 of the cell cycle, the cytoplasm and cell membrane split, resulting in the creation of 2 identical daughter cells. Stem cells. A stem cell is a unique type of cell that can develop into various specialized cells. The stem cells are undifferentiated in multicellular organisms. The 2 types of stem cells in mammals are adult stem cells and embryonic stem cells. Location. Adult stem cells are primarily located in the bone marrow of both adults and children. The embryonic cells are typically located within the inner cell mass of a developing embryo known as a blastocyst. Differentiation. Adult stem cells can differentiate to form only certain types of cells. For example, the stem cells in the bone marrow are limited to differentiating into specific types of blood cells. Embryonic cells can develop into any specialized cells of the body like nerve cells, muscle cells, and more. Advantages. The advantages of the adult stem cells are as follows. Using adult stem cells in medical treatments is safe, as donors recover quickly. The use of adult stem cells presents fewer ethical concerns. Patients with leukemia may undergo a stem cell transplant to potentially restore their bone marrow to a healthy state. The advantages of embryonic stem cells are as follows. Since embryonic stem cells can develop into any specialized cell, they can treat many diseases. It is possible to extract embryonic stem cells from unused embryos at fertility clinics. It might be possible to grow whole replacement organs using embryonic stem cells. Limitations. There are some limitations in adult stem cells. Finding a suitable donor for adult stem cells might take a long time. It can treat only a few diseases, as they can only differentiate into specific types of cells. Likewise, embryonic stem cells do have some limitations. The use of embryonic cells for medical purposes is not ethically permissible since every embryo has the potential to become alive. There is a risk of viral infection for patients undergoing treatment with embryonic stem cells, as this is a relatively new form of treatment. Plant meristems. Meristem is a tissue composed of undifferentiated stem cells capable of cell division and growth. They are usually found on the roots and shoots in plants. Meristem tissues are the building blocks of the specialized plant structures. The meristem cells can differentiate into all types of plant cells. Therefore, they can be used to clone the entire plant. Cloning creates precise replicas of an organism, cell, or DNA sequence. Plant meristems can also be used to clone plants with unique features or medicinal values to produce multiple copies. Cloning can be a helpful solution in preventing rare species from going extinct. They are a cost-effective way to produce a vast quantity of plants. When plants are cloned, any diseases or genetic flaws present in the original plant will be replicated in all the cloned plants, which can ultimately lead to the demise of all the plants. Therapeutic cloning. Therapeutic cloning involves extracting stem cells from cloned embryos without ever implanting them in a womb. It is also called somatic cell nuclear transfer. This type of cloning produces stem cells with identical genes to the patient, making them suitable for medical treatments. It can also be used for growing new organs and transplants. Therapeutic cloning does not involve implanting embryos in wombs.